Very excited today to introduce Dr. Rupangi C. Vasavada. She is coming to us from the City of Hope in Duarte, California, which is just outside of Los Angeles. And she's going to present a talk called Clinical Trial of Denosumab for Type 1 Diabetes. Very interesting new clinical trial, and we're hoping to get newly diagnosed patients between 18 and 50 interested in joining this very exciting opportunity, this clinical trial. And a little bit about Dr. Vasavada. She's an associate professor in the Department of Translational Research and Cellular Therapeutics at City of Hope. She received her PhD in molecular biology from UPenn and Master of Science in Zoology from Delhi University in India. She was at the Icahn School of Medicine at Mount Sinai, New York, and at the University of Pittsburgh prior to joining City of Hope. Her research is centered on understanding the pathophysiology of diabetes and finding ways to treat the disease. Primarily, her research is focused on proteins and pathways that can enhance the preservation and regeneration of functional pancreatic beta cells. And recent work in her laboratory has been directed towards understanding the role of extracellular vesicles as functional and molecular biomarkers in type 1 diabetes. Her work's been supported over the years with grants from JRF, now Breakthrough, and ADA, NIH, where she serves as a member of their study section to review grants. And her talk today is going to really, illustrate this opportunity and the science behind it. Thank you so much, Dr. Vasavada, for joining us, and excited to hear what you have to share.
Thank you. Can you hear me? You can,
Yes,
right?
perfect.
Okay. Thank you very much, Monica, for this opportunity to talk about the clinical trial. And thanks for whoever's on the call to hear the talk. Thank you for joining. So today I'm going to be talking about the clinical trial of Denosumab for type 1 diabetes that's ongoing at 3 different sites, as you can see here. And this basically, the trial is funded by Breakthrough T1D, or formerly known JDRF. And we've also been fortunate to get supplemental funds from the Warnick Family Project for Type 1 Diabetes at City of Hope for this trial. Now, before I talk about the trial, I would just like to give a brief sort of, how we got where we are today and give a little bit of introduction on that. So, as you know, type 1 diabetes is an autoimmune disorder characterized by immune cell-mediated destruction of insulin-producing beta cells. This is a pancreatic islet. This is a beta cell and when the beta cell gets stressed, it starts releasing proteins, chemokines that basically attract immune cells from the local environment. And these immune cells themselves start producing the proteins like cytokines and chemokines that damage the islet and the beta cell and cause beta cell death. This leads to glucose dysregulation and lifelong dependence on exogenous insulin to survive. Now, there are therapeutic interventions that are ongoing. Most of them have been for modulation of the immune system, which is extremely important. And also, we need to improve beta cell health, but there are fewer clinical trials on improving beta cell health. Most of them so far have been focused on the immune system. Now, what I'm going to talk about today, the effect of Denosumab, is mainly, we believe, on beta cell health. It could also maybe modulate the immune system, but we do know that it modulates the beta cell health. So question is, what is Denosumab? So Denosumab is actually a medication that is used to treat osteoporosis and bone loss, and also bone tumors, as shown here. This is the package for Denosumab. It has been FDA approved in the US since 2010, so it's over 15 years now, and is being used by millions of people worldwide. It's currently marketed as 2 different brand names, Prolia, as shown here, which is given as a subcutaneous injection, 60 milligrams every 6 months for osteoporosis. They also have the same Denosumab is packaged as Xgeva, which is double the dose, 120 milligrams, and it's given every month for cancer-related bone events. More severe bone events are given Xgeva. And both have been approved since 2010. So question is, how does Denosumab work? So Denosumab works by inhibiting this pathway, the rank ligand pathway. So rank is a receptor on the bone that's present on the bone, which interacts with this protein rank ligand. And the two together, activation of this pathway causes events inside the bone that leads to bone thinning or osteoporosis. Now, scientists at Amgen actually wanted to stop this process, and so they developed this drug, Denosumab, which basically acts by binding this protein, rank ligand, acts like a sponge, inhibits its action on this pathway, and therefore it stops bone thinning. So this, and that's why this drug has been developed for osteoporosis. So what does this have to do with the beta cell or the islet or diabetes? So over the years, my lab actually showed that this pathway, the rank ligand pathway, is present on the islet and is present on the beta cell. And what we showed, and others have also shown this, is that many stressors that are related to type 1 and type 2 diabetes, for example, high glucose, high lipids, cytokines, pro-inflammatory cytokines, all of them increase this expression of these proteins on the beta cell and increase activation of this pathway. Now, we found over the years that this pathway is bad for the beta cell. It decreases the growth of the pancreatic beta cell, it increases death, and decreases function. And we showed that over the years using multiple different methods, as shown here. There are these 3 independent ways that we showed that this pathway is bad for the beta cell. We removed this receptor rank, as shown here, deleted it. We also used 2 different proteins that actually inhibit this pathway. One is Denosumab that I just talked about, which actually binds this rank ligand to inhibit it. And we also used a naturally occurring protein called OPG, which also binds rank ligand and inhibits this pathway. So by using 3 independent methods in mouse and human islets in culture, we showed that by inhibiting this pathway, we can improve beta cell health. We can increase the growth, decrease the death, and increase the function of these cells. Now, furthermore, we wanted to see if it works in vivo, in an animal model. So to do that, we basically took mice that spontaneously developed type 1 diabetes. These are the NOD mice. And we injected OPG into this mice. Now, we could not inject Denosumab because Denosumab is specific to human rank ligand, so it won't work in mice. So we used the next best thing, which was OPG, which does act on the mouse rank ligand. So here, when we injected these mice, after they get diabetes, we allowed these mice to get diabetes. As you can see here, their blood sugar is in the 350 milligrams per deciliter range. Then we injected these mice either with OPG or with 2 other control molecules, vehicle or IgG. You can see that only the mice that got OPG had decreased blood sugar levels. The other 2 groups increased their blood sugar levels over time. And when you look at diabetes incidence, you can see that the OPG-treated mice, we had a reversal in diabetes by 70%, but not in the others. So based on all of this data, we believe that Denosumab has potential for diabetes. As I've told you, that this rank ligand pathway in bone induces bone thinning. I've also showed you that this pathway in the islet and the beta cell is bad for the beta cell. Now, others in the field, other scientists have also shown that this pathway is present in the liver, muscle, and fat, which are the insulin sensing tissues. And they've shown that in mouse models of type 2 diabetes, activation of this pathway causes insulin resistance. And our data has also, in vitro data has shown that activation of this pathway in the immune cells causes immune cell inflammation. So based on all of this, we believe that Denosumab, which actually has been used for this bone osteoporosis, by blocking this pathway, should have a similar beneficial effects on the islet, on these insulin-sensing tissues, as well as on immune cell inflammation, and may have potential for both type 1 and type 2 diabetes. So based on our findings, actually, Breakthrough T1D funded us for this clinical trial. And the aims of this clinical trial are to see if Denosumab can preserve or boost beta cell health and allow people with early type 1 diabetes to continue to make their own insulin longer. So the initial clinical trial will determine if Denosumab is safe and effective in improving beta cell function, because this has not been tested in people with type 1 diabetes before who have likely no osteoporosis. So we will also examine whether Denosumab causes any bone changes because it is a bone drug. Now, what is the study design? It's a phase 1-2 clinical trial. 1 is for safety, 2 is for efficacy. It's a prospective, randomized, double-blinded, placebo-controlled, multi-center trial, which I will go through. It is the indications for adults with early type 1 diabetes. This is 1 to 5 years since diagnosis, who have residual beta cell function that we examined by presence of C-peptide. And this is important because we believe that Denosumab should have an impact on the beta cell. So we do need to make sure that the people have residual beta cells. Study sites, there are 3 study sites involved in this trial. City of Hope in California is via the lead site. We are partnering with University of Alabama at Birmingham, as well as Indiana University. Our objective is to recruit 45 subjects. This is an open and competitive recruitment across all sites. And we are going to have 2 treatment groups. So subjects will be randomized at a 2-to-1 ratio, which means that 30 people will get treatment, 15 will get the placebo. So there are going to be 2 treatment groups. The first group will get Denosumab, and this will be at the 60 milligram dose, as I showed you, which is present in Prolia, and it will be given subcutaneously under the skin. But instead of giving every 6 months, we are going to give it every 3 months. So we are going to give, and this is over a period of 1 year. So they're going to be a total of 4 injections over 1 year. The other group will be saline, placebo group that will get saline in the same regimen as we'll be giving the Denosumab. And the follow-up, as I said, it's a 1-year study. So at 12 months, we'll have the final follow-up after the first injection. As I told you, there are 3 participating sites. The City of Hope in California is a lead site, myself and Dr. Kandil, who is actually the principal investigator of the clinical trial, are conducting the site. Jeanette Hacker is actually our project manager. She manages the whole clinical trial, and she herself is a long-term type 1 diabetes patient and is very, very involved in all the clinical trials ongoing. Then we have Dr. Carmela Evans Molina at Indiana University, who has been involved in multiple type 1 diabetes clinical trials over the years. And Dr. Annette Shalev at University of Alabama at Birmingham, and she also has been involved in other clinical trials with type 1 diabetes. So again, as you can see, that we have 3 sites at different parts of the country. So what is the study timeline? So basically, we are going to have the patients from 18 to 50 years old diagnosed within 1 to 5 years with a positivity of C-peptide. They first signed informed consent. Then we have a first pre-screening visit where only the C-peptide and vitamin D will be measured. Then we have a second visit, the screening visit, where they will be screened from mixed meal tolerance test to see whether the C-peptide is 6.6 nanogram per ml or more. And other aspects will be examined here. And if the patients qualify with all the inclusion-exclusion criteria, they come in for their first visit for a treatment, which is considered the 0 day. And then every 3 months, they will be coming for the treatment. And at the end, by 12 months, they will come for the final visit checkup. Now, what are the inclusion criteria? So we are looking for adults, females 18 to 50 years of age, and males 21 to 50 years of age. And the difference in this initial age is because of the minimum age that's based on skeletal maturity. So we know that females' skeletal maturity occurs earlier compared to the males, and that's why we have this age group. The duration of type 1 diabetes, we've chosen to be of 1 to 5 years. And why is that? Because we're hoping that we know that there's a honeymoon period in the initial phase of type 1 diabetes, and to avoid any, impact of the honeymoon period on the study, we chose, after 1 year. And 5 years is because it's known that the C-peptide positivity, there are sufficient numbers of people within that 1 to 5 year window. So that's why we chose that. Of course, they should have one or more diabetes autoantibodies. As I mentioned earlier, they should have residual beta cell function, which is C-peptide positivity, normal calcium levels, and of course, should be using effective method of birth control during and a few months after the study. What are the exclusion criteria? Main exclusion criteria is use of other investigational agents within 3 months of enrollment, other medications besides insulin within 3 months of enrollment, vitamin D3 deficiency, high BMI, history of delayed puberty, high HbA1c, any hypoglycemic and DKA events within 3 months prior to screening. We're also looking for not having bone fractures within the past 12 months and dental or oral disease. And this is coming because of the effects of denosumab on these things. So we want to make sure that there is no, issue starting out. And it's known that in denosumab and in osteoporosis, when people stop taking denosumab, they're more prone to getting bone fractures. So these are people who already have osteoporosis. So the increase in bone density that occurs during denosumab treatment goes down after they withdraw from denosumab, and therefore they become more susceptible to bone fractures. So we want to make sure that there are none of these events starting out. Of course, pregnancy or active breastfeeding is also key exclusion criteria. So what is the study treatment plan? As I said, there'll be 2 groups, one group with denosumab given 60 milligrams every 3 months and placebo given saline in the same regimen. The drug will be administered by a study nurse during an outpatient clinic. It will be injected under the skin, either on the upper arm, upper thigh, and or abdomen. And both investigators double-blinded. That means investigators, the PIs who are conducting the study, as well as the subjects who are getting the treatment, are blinded to the treatment. So we don't know till after. We will also be supplementing the patients with calcium and vitamin D, because denosumab has been associated with causing low blood calcium levels. To avoid any complications, patients will be given oral calcium and vitamin D3 supplements. We expect that insulin therapy and blood glucose monitoring will continue, but no other injectable diabetes medications other than insulin will be there. And then we also would expect avoid major changes in insulin delivery mechanism, diet, exercise, and other behaviors. The follow-up assessment. So there'll be 3 specific follow-up assessments every 3 months: safety and adverse events, effectiveness of the drug on beta cell function, and bone health. And how are we going to do that? For the safety, again, as during each injection, the patient will be monitored for at least 15 minutes to make sure there's no hypersensitive reaction. They will be provided contact information with the clinical study nurse for 24 hours for safety concerns. And 1 week after each dose, the nurse will call to follow up to find out what's happening. And regular safety visits at every 3 months. We will be reviewing symptoms, vitals, blood glucose monitoring, physical exams, labs, and any other adverse events happening. In terms of the efficacy, to see whether denosumab is doing what we hope it will do, which is the main primary efficacy is going to be the mixed meal tolerance test, which is where the person is given a boost drink, and then we measure take blood every 30 minutes before and after, and measure various hormones, C-peptides, etc., to understand what's happening to their beta cell function. So these are the things we'll be measuring. We'll also look at fasting C-peptide, glucose, insulin, glucagon, etc. We'll also measure the HbA1c, which is the glucose levels for the 3 months, and that will give us an indication of their glucose control. The continuous glucose monitoring will be evaluated. We'll evaluate the variability in the blood glucose, that analyze the time in range, any hypoglycemia events, etc., will be monitored. And the daily insulin requirement, which that's logged in, will also be monitored and looked at. So in terms of bone health assessments, we will, because denosumab impacts the bone, we will look for blood bone markers, and they will be evaluated at baseline, 6 and 12 months. And we'll also look at the bone density by doing a DEXA scan before the patient starts the treatment and 12 months after. Now, we are also going to compensate and reimburse the participants in the study. So they will be compensated, as you can see, for a total of $900 if they finish the 1-year study, and it's broken down as shown here based on each visit and what is being done at the visit. And besides this, we will also be reimbursing the patients for their travel costs, food-related costs and other expenses. So what are the potential risks? Now, these are all out there because denosumab has been used for 15 years. So if you look up the internet, you'll see all of these things. So the most common adverse effects are back and joint pains. In women, elevated cholesterol, hypertension, headache, cold symptoms. There are rare but serious side effects that have happened. That includes hypersensitivity. When it's injected, is there any hypersensitivity to the drug? Because as I told you earlier, it causes low calcium levels. So we are using calcium and vitamin D3 supplements for that. There is effects on osteonecrosis of the jaw sometimes. And as I said, bone fractures after the stopping of denosumab in patients with osteoporosis. They also have severe bone joint muscle pains. And because it impacts immune system, the people have more prone to certain infections. That has been shown. What are the potential advantages and future directions? So we are repurposing an FDA-approved drug. So we believe it should be easier. If it works, it should be easier to market for a new indication. It's been widely used for over 15 years, so we believe that it's a much better, well-established, relatively safety profile has been established. It's injected subcutaneously like insulin versus IV infusions like some other drugs. Also, it has a very long half-life. So as in osteoporosis, every 6 months, we are planning every 3 months for various reasons. But it therefore allows infrequent doses. You only need to take it every 3 months or so. The potential effects, beneficial effects, we believe will be on the long run on beta cell function, beta cell health, beta cell number may increase is what we believe based on everything we have found in the lab. And it may even have a positive impact on the inflammatory response besides the beta cell. Now, if this initial trial is successful, future studies could explore extended dosing and denosumab use at other stages of type 1 diabetes and, of course, other diabetes types like type 2 diabetes. So with that, I just want to sort of say we are looking for volunteers for the study. And there are 3 sites. These are the contact information. And this is also being posted on clinicaltrials.gov as shown here. I would also actually like to thank the people who have done this work over the years and how we got where we got. People in my lab over the years. Jeanette, as I said, she's a project manager with Dr. Kandil. These are the people currently in my lab, and Nagesh is the person whose work has actually resulted in this denosumab coming to clinical trials in type 1 diabetes, with help from all the other people in the lab over the years. Thank you to all the funding agencies, all our collaborators over the years, and most of all, to the volunteers and the donors of human samples. I couldn't do anything without that. So I will stop here. Just this is, if anybody's interested and if we want to pass the message around, where we can be contacted, here it is. So
Thank you so much for this really great information, Ruby. I'm just opening up for questions. You can feel free to unmute yourself and ask your question or drop a question in the chat and we will read it aloud for everyone. So far there are 3 patients enrolled, correct?
that's correct.
And there's
3 treated and enrolled, and we are hoping another 3 to 5.
So then you're at 8. So then you would need about 20 or so more patients to who will actually be taking the medication, and then you have your other cohort. I think you said 15 would be the placebo.
So 30 for the treatment, 15 for the placebo. And of course, it's all blinded. So we don't know who's getting what or when. So,
Right.
yes.
And then I guess could you unpack a little bit? You did tap on some of this, bone fracture and so forth, but that's really pertaining to people who were treated for osteoporosis. So
Right.
in this cohort, it's like age 19 to 50, and most of those people are really not going to be at risk for osteoporosis at this point in life. Is that
Exactly. That's correct. I mean,
so?
that's what we think. So, and we looked, is there anything in the Amgen literature? And because when they were developing the drug, what have they found? Anything on people without osteoporosis? What happens? But there's nothing out there. We tried looking hard, didn't
Mhm.
find anything. But like you said, and again, we are making sure the patients have all the information and,
Yeah,
all of the dental thing. But based on everything I'm seeing on the literature, even with osteoporosis, what they say is that the bone density increases, and once you stop, it goes down, and it goes down to a level sort of where they were, maybe a little less, but usually where they were. And because they already have osteoporosis, the bone fracture risk, of course, increases.
right,
Do believe that that's unlikely in this patient population because they don't
right.
have osteoporosis to begin with, most likely. So,
Yeah, I just wanted to kind of highlight that even
yes,
though it does perhaps is a little bit like a little more daunting, it may not pertain to you.
exactly.
So it might
Exactly.
be worth sort of wading into the waters and getting more information to see. And ultimately, if you are newly diagnosed and you have some residual beta cell function, there's kind of a good possibility that you could protect and preserve beta cell function going forward. So that's the optimal outcome.
Yes, that's what we are hoping for based on, I mean, so I just, the fact that I know this is a mouse model, et cetera, but the fact that it reversed diabetes in mice, to me, that tells me it's not just the beta cell it is acting on, it also has to act on the immune system in a positive
Right.
way for that to happen. And
Yep.
in collaboration with Helena's group and Jay, we've shown that in culture, in human immune cells, it does reduce inflammation. So there is hope there too, but we don't know for sure. So that's
Yeah.
something we would like to look at. Actually, the blood draws we'll have, we are going to look at inflammation markers to see if it has any impact on the immune system. So that's something we'd love to see.
So while you're doing something potentially very positive for your own health, you're also contributing to the knowledge base in the scientific realm of type 1
that's right.
diabetes. It's a
That's
win-win
very true.
situation.
Very true. Yes.
Yeah, so I guess if there are no questions, we're at the end of our 30 minutes. Beautiful work. Thank you so much.
Sure.
There's a question, wait, from George Harb. Beautiful work, Ruby. Thank you. Great to see you. Have you considered
Oh,
an earlier T1D onset patient population, less than 1 year onset, and combining with teplizumab? Great question.
great question. Actually, to be honest, this whole thing started as a little innovative grant I wrote for JDRF at that time, doing combination with this and anti-CD3. They didn't think that, but so they came back saying you should be looking at denosumab in type 1 diabetes people based on everything you've shown. So that is definitely something that we will be looking into, but step at a time. But yes,
Right.
completely agree, George. Hi, good to see you here. Not see you, but at least see you.
the
Yeah.
audience. Yeah,
Yes.
it's great to have, that's a really great point to bring up, that there's another, if this proves to be effective, that's yet another tool in the toolbox for clinicians to have when they are thinking about multi-drug treatment of type 1
Mhm.
diabetes. And I would just say, because City of Hope is a big cancer center, if we think about how the breast cancer treatment evolved, um, it's a good playbook for, type 1 diabetes treatment to sort of consider. As at first, many years ago, 20, 30 years ago, if you had breast cancer, you just had one medication in your arsenal. And now there's a whole host of medications. It's very personalized medicine. And so, in developing these other alternative medicines, to be either used in combination or on their own in different windows of time, um, it will really strengthen the treatment protocol available to those who have type 1.
Completely agree. There's not going to be one treatment for anything, right? It has to be multiple treatments, and the more you have, the more you bandwidth you'll have to treat the patients. Basically, I think so. Yeah.
Yeah, more tools in the toolbox,
Exactly. Exactly.
the
Exactly.
better you can address the disease. So
Yes.
with that hope in mind, thank you all for participating, and I hope this encourages you to reach out and or share this presentation with those who it may help. This presentation will be posted on our video library. It's available through our website, www.thesugarscience.org, or you can go and search us directly, The Sugar Science, on YouTube, and we'll have it there. So
Great.
thank you again, Dr.
Thank
Vasavada,
you.
and
Thank you, everyone, for coming, and thank you, Monica, for this opportunity. Really appreciate it.
of course. Thanks, and have a great rest of your day.
Thank you. Bye
Bye-bye.
bye.